Dna Nanotechnology is the design and manufacture of artificial nucleic acid structures for technological uses. In this field, nucleic acids are used as non-biological engineering materials for nanotechnology rather than as the carriers of genetic information in living cells. Researchers in the field have created static structures such as 2- and 3-dimensional crystal lattices, nanotubes, polyhedra, and arbitrary shapes, as well as functional devices such as molecular machines and DNA computers. The field is beginning to be used as a tool to solve basic science problems in structural biology and biophysics, including applications in crystallography and spectroscopy for protein structure determination. Potential applications in molecular-scale electronics and nanomedicine are also being investigated. The conceptual foundation for Dna nanotechnology was first laid out by Adrian Seaman in the early 1980s, and the field began to attract widespread interest in the mid-2000s. This use of nucleic acids is enabled by their strict base pairing rules, which cause only portions of strands with complementary base sequences to bind together to form strong, rigid double helix structures. This allows for the rational design of base sequences that will selectively assemble to form complex target structures with precisely controlled, nanoscale features. A number of assembly methods are used to make these structures, including tile-based structures that assemble from smaller structures, folding structures using the DNA Origami Method, and dynamically reconfigurable structures using Strand displacement techniques. While the field name specifically references DNA, the same principles have been used with other types of nucleic acids as well, leading to the occasional use of the alternative name nucleic acid nanotechnology. Fundamental concepts. Properties of nucleic acid nanotechnology is often defined as the study of materials and devices with features on a scale below 100 nanometers. Dna nanotechnology, specifically, is an example of bottom-up molecular self-assembly, in which molecular components spontaneously organize into stable structures. The particular form of these structures is induced by the physical and chemical properties of the components selected by the designers. In Dna nanotechnology, the component materials are strands of nucleic acids such as DNA. These strands are often synthetic and are almost always used outside the context of a living cell. DNA is well suited to nanoscale construction because the binding between two nucleic acid strands depends on simple base pairing rules which are well understood, and form the specific nanoscale structure of the nucleic acid double helix. These qualities make the assembly of nucleic acid structures easy to control through nucleic acid design. This property is absent in other materials used in nanotechnology, including proteins, for which protein design is very difficult, and nanoparticles, which lack the capability for specific assembly on their own. The structure of a nucleic acid molecule consists of a sequence of nucleotides distinguished by which nucleobase they contain. In DNA, the 4 bases present are adenine, cytosine, guanine, and thymine. Nucleic acids have the property that 2 molecules will only bind to each other to form a double helix if the 2 sequences are complementary, meaning that they form matching sequences of base pairs, with A only binding to T, and C only to G. Because the formation of correctly matched base pairs is energetically favorable, nucleic acid strands are expected in most cases to bind to each other in the conformation that maximizes the number of correctly paired bases. The sequences of bases in a system of strands thus determine the pattern of binding and the overall structure in an easily controllable way. In Dna nanotechnology, the base sequences of strands are rationally designed by researchers so that the base pairing interactions cause the strands to assemble in the desired conformation. While DNA is the dominant material used, structures incorporating other nucleic acids such as RNA and peptide nucleic acid have also been constructed. Subfield DNA nanotechnology is sometimes divided into two overlapping subfields: structural DNA nanotechnology and dynamic DNA nanotechnology. Structural DNA nanotechnology, sometimes abbreviated as SDN, focuses on synthesizing and characterizing nucleic acid complexes and materials that assemble into a static, equilibrium end state. On the other hand, dynamic DNA nanotechnology focuses on complexes with useful non-equilibrium behavior, such as the ability to reconfigure based on a chemical or physical stimulus. Some complexes, such as nucleic acid nanomechanical devices, combine features of both the structural and dynamic subfields. The complexes constructed in structural DNA nanotechnology use topologically branched nucleic acid structures containing junctions. One of the simplest branched structures is a 4-arm junction that consists of 4 individual DNA strands, portions of which are complementary in a specific pattern. Unlike in natural holiday junctions, each arm in the artificial immobile 4-arm junction has a different base sequence, causing the junction point to be fixed at a certain position. Multiple junctions can be combined in the same complex, such as in the widely used double crossover motif, which contains 2 parallel double helical domains with individual strands crossing between the domains at 2 crossover points. Each crossover point is itself topologically a 4-arm junction, but is constrained to a single orientation, as opposed to the flexible single 4-arm junction, providing a rigidity that makes the DX motif suitable as a structural building block for larger DNA complexes. Dynamic DNA nanotechnology uses a mechanism called Toehold mediated strand displacement to allow the nucleic acid complexes to reconfigure in response to the addition of a new nucleic acid strand. In this reaction, the incoming strand binds to a single-stranded toehold region of a double-stranded complex, and then displaces one of the strands bound in the original complex through a branch migration process. The overall effect is that one of the strands in the complex is replaced with another one. In addition, reconfigurable structures and devices can be made using functional nucleic acids such as deoxyribozymes and ribozymes, which are capable of performing chemical reactions, and aptamers, which can bind to specific proteins or small molecules. Structural DNA nanotechnology. Structural DNA nanotechnology, sometimes abbreviated as SDN, focuses on synthesizing and characterizing nucleic acid complexes and materials where the assembly has a static, equilibrium endpoint. The nucleic acid double helix has a robust, defined 3-dimensional geometry that makes it possible to predict and design the structures of more complicated nucleic acid complexes. Many such structures have been created, including 2- and 3-dimensional structures, and periodic, aperiodic, and discrete structures. Extended lattices. Small nucleic acid complexes can be equipped with sticky ends and combined into larger 2-dimensional periodic lattices containing a specific tessellated pattern of the individual molecular tiles. The earliest example of this used double crossover complexes is the basic tiles, each containing 4 sticky ends designed with sequences that cause the DX units to combine into periodic 2-dimensional flat sheets that are essentially rigid 2-dimensional crystals of DNA. 2-dimensional arrays have been made from other motifs as well, including the holiday junction rhombus lattice, and various DX-based arrays making use of a double cohesion scheme. The top 2 images at right show examples of tile-based periodic lattices. 2-dimensional arrays can be made to exhibit aperiodic structures whose assembly implements a specific algorithm, exhibiting one form of DNA computing. The DX tiles can have their sticky end sequences chosen so that they act as Wang tiles, allowing them to perform computation. A DX array whose assembly encodes an XOR operation has been demonstrated. This allows the DNA array to implement a cellular automaton that generates a fractal known as the Sierpinski gasket. The third image at right shows this type of array. Another system has the function of a binary counter, displaying a representation of increasing binary numbers as it grows. These results show that computation can be incorporated into the assembly of DNA arrays. DX arrays have been made to form hollow nanotubes 4 to 20 nanometers in diameter, essentially two-dimensional lattices which curve back upon themselves. These DNA nanotubes are somewhat similar in size and shape to carbon nanotubes, and while they lack the electrical conductance of carbon nanotubes, DNA nanotubes are more easily modified and connected to other structures. One of many schemes for constructing DNA nanotubes uses a lattice of curved DX tiles that curls around itself and closes into a tube. In an alternative method that allows the circumference to be specified in a simple, modular fashion using single-stranded tiles, the rigidity of the tube is an emergent property. The creation of three-dimensional lattices out of DNA was the earliest goal of Dna Nanotechnology, but this proved to be one of the most difficult to realize. Success using a motif based on the concept of tensegrity, a balance between tension and compression forces, was finally reported in 2009. Discrete structures researchers have synthesized a number of three-dimensional DNA complexes that each have the connectivity of a polyhedron, such as a cube or octahedron, meaning that the DNA duplexes trace the edges of a polyhedron with a DNA junction at each vertex. The earliest demonstrations of DNA polyhedra were very work-intensive, requiring multiple ligations and solid phase synthesis steps to create catenated polyhedra. Subsequent work yielded polyhedra whose synthesis was much easier. These include a DNA octahedron made from a long single strand designed to fold into the correct conformation, and a tetrahedron that can be produced from 4 DNA strands in a single step, pictured at the top of this article. Nanostructures of arbitrary, non-regular shapes are usually made using the DNA Origami method. These structures consist of a long, natural virus strand as a scaffold, which is made to fold into the desired shape by computationally designed short, staple strands. This method has the advantages of being easy to design, as the base sequence is predetermined by the scaffold strand sequence, and not requiring high strand purity and accurate stoichiometry, as most other DNA nanotechnology methods do. DNA origami was first demonstrated for two-dimensional shapes, such as a smiley face and a coarse map of the Western Hemisphere. Solid three-dimensional structures can be made by using parallel DNA helices arranged in a honeycomb pattern, and structures with two-dimensional faces can be made to fold into a hollow overall three-dimensional shape, akin to a cardboard box. These can be programmed to open and reveal or release a molecular cargo in response to a stimulus, making them potentially useful as programmable molecular cages. Templated assembly nucleic acid structures can be made to incorporate molecules other than nucleic acids, sometimes called heteroelements, including proteins, metallic nanoparticles, quantum dots, and fullerenes. This allows the construction of materials and devices with a range of functionalities much greater than is possible with nucleic acids alone. The goal is to use the self-assembly of the nucleic acid structures to template the assembly of the nanoparticles hosted on them, controlling their position and in some cases orientation. Many of these schemes use a covalent attachment scheme, using oligonucleotides with amide or thiol functional groups as a chemical handle to bind the heteroelements. This covalent binding scheme has been used to arrange gold nanoparticles on a DX-based array, and to arrange streptavidin protein molecules into specific patterns on a DX array. A non-covalent hosting scheme using Dervan polyamides on a DX array was used to arrange streptavidin proteins in a specific pattern on a DX array. Carbon nanotubes have been hosted on DNA arrays in a pattern allowing the assembly to act as a molecular electronic device, a carbon nanotube field effect transistor. In addition, there are nucleic acid metallization methods, in which the nucleic acid is replaced by a metal which assumes the general shape of the original nucleic acid structure, and schemes for using nucleic acid nanostructures as lithography masks, transferring their pattern into a solid surface. Dynamic DNA nanotechnology. Dynamic DNA nanotechnology focuses on creating nucleic acid systems with designed dynamic functionalities related to their overall structures, such as computation and mechanical motion. There is some overlap between structural and dynamic DNA nanotechnology, as structures can be formed through annealing and then reconfigured dynamically, or can be made to form dynamically in the first place. Nanomechanical devices. DNA complexes have been made that change their conformation upon some stimulus, making them one form of nanorobotics. These structures are initially formed in the same way as the static structures made in structural DNA nanotechnology, but are designed so that dynamic reconfiguration is possible after the initial assembly. The earliest such device made use of the transition between the bDNA and zDNA forms to respond to a change in buffer conditions by undergoing the twisting motion. This reliance on buffer conditions, however, caused all devices to change state at the same time. Subsequent systems could change states based upon the presence of control strands, allowing multiple devices to be independently operated in solution. Some examples of such systems are molecular tweezers, design that has an open and a closed state, a device that could switch from a paranemic crossover conformation to a double junction conformation, undergoing rotational motion in the process, and a two-dimensional array that could dynamically expand and contract in response to control strands. Structures have also been made that dynamically open or close, potentially acting as a molecular cage to release or reveal a functional cargo upon opening. DNA walkers are a class of nucleic acid nanomachines that exhibit directional motion along a linear track. A large number of schemes have been demonstrated. One strategy is to control the motion of the walker along the track using control strands that need to be manually added in sequence. Another approach is to make use of restriction enzymes or deoxyribozymes to cleave the strands and cause the walker to move forward, which has the advantage of running autonomously. A later system could walk upon a two-dimensional surface rather than a linear track, and demonstrated the ability to selectively pick up and move molecular cargo. Additionally, a linear walker has been demonstrated that performs DNA templated synthesis as the walker advances along the track, allowing autonomous multi-step chemical synthesis directed by the walker. The synthetic DNA walker's function is similar to that of the proteins dynein and kinesin. Strand displacement cascades. Cascades of strand displacement reactions can be used for either computational or structural purposes. An individual strand displacement reaction involves revealing a new sequence in response to the presence of some initiator strand. Many such reactions can be linked into a cascade where the newly revealed output sequence of one reaction can initiate another strand displacement reaction elsewhere. This in turn allows for the construction of chemical reaction networks with many components, exhibiting complex computational and information processing abilities. These cascades are made energetically favorable through the formation of new base pairs, and the entropy gain from disassembly reactions. Strand displacement cascades allow for isothermal operation of the assembly or computational process, as opposed to traditional nucleic acid assemblies requirement for a thermal annealing step, where the temperature is raised and then slowly lowered to ensure proper formation of the desired structure. They can also support catalytic functionality of the initiator species. where less than one equivalent of the initiator can cause the reaction to go to completion. Strand displacement complexes can be used to make molecular logic gates capable of complex computation. Unlike traditional electronic computers, which use electric current as inputs and outputs, molecular computers use the concentrations of specific chemical species as signals. In the case of nucleic acid strand displacement circuits, the signal is the presence of nucleic acid strands that are released or consumed by binding and unbinding events to other strands in displacement complexes. This approach has been used to make logic gates such as AND or NOT gates. More recently, a 4-bit circuit was demonstrated that can compute the square root of the integers 0 to 15, using a system of gates containing 130 DNA strands. Another use of strand displacement cascades is to make dynamically assembled structures. These use a hairpin structure for the reactants, so that when the input strand binds, the newly revealed sequence is on the same molecule rather than disassembling. This allows new opened hairpins to be added to a growing complex. This approach has been used to make simple structures such as 3- and 4-arm junctions and dendrimers.